Hi everyone, my name is Samantha Landrer. I'm a third-year student in the Genetics and Molecular Biology program here at Emory University, and I'm currently in the Whitehead Biomedical Research Building on the third floor within the Department of Human Genetics. And let me go show you my lab. Nobody's in lab right now, but in the middle we have all of our research bays, and then on the outside is where we have all of our seats, where all of the students and everyone that works in the lab sit. Since nobody is in lab, I can take my mask off for a little bit, just so I can show you all around. In my lab, we study glial development, specifically of astrocytes, which are the most numerous glia cells within the central nervous system. So we want to know how they develop, how they mature, and what various functions there are in the brain that may contribute to neurodevelopmental disorders. So the way we do this is through the use of organoids. I'll explain it more, so I'm going to take you guys into our stem cell room, but first I gotta gear up. It's one of our more sterile rooms, so we have to try to put on as much protection as possible. Okay, I got my hair up, I got my lab coat on, and my gloves on also that have been ethanol. So in the incubators behind me, we have human cortical organoids. So what these are is that they're induced pluripotent stem cells that are formed into 3D balls and then patterned in order to differentiate into cell types of the brain. So within our organoid system, we have radioglia, which are progenitor cells of the brain, neurons, and astrocytes. So I'm going to show you what they look when they're just formed, when they're little babies, and then how they look when they're older. These organoids right here were formed only a few days ago, so they kind of look like messy blobs right now, but in a few days, they'll all look a little bit more like this. Here's a plate of organoids that are about a month and a half older than the ones I just showed you, and you can see how clearly you can see them in the dish now. And they look like little couscous, to be honest. And when we look at them under the microscope, you can see that they form nice and circular spheres, and they're also a lot bigger than they were before. So we have a lot of them in the dish, and they're all pretty rounded.
Now here's for the cool part. We've labeled our organoids with a GFP virus that specifically labels neurons. So it uses the human synapsin promoter, and now you're able to see all of these amazing projections from neurons. And the reason it's hard to really get focus is just because it's 3D tissue. So there's so many layers that it's really hard to focusing in on a single layer. But you can see still as we go throughout the organoid, there's projections going all throughout. You can see the cell body. Now for the best part are our astrocytes. So we similarly labeled our organoids with a virus that is specific to astrocytes using a GFAP mCherry. So once again, you can see all the astrocytes throughout the organoids. You can see a bunch of projections and how they just span all throughout the organoid. And then we can actually isolate our our cell types from the organoids. And you can see here that we have in the cyan our astrocytes, and in the yellow, or in the next picture in the gray, are our neurons. So it's really cool that we're able to pull out these cell types and then analyze them even further. You can see all of the synapses and all of the amazing projections. And then lastly, I just want to say thank you for letting me show our lab to you for this year's Brain Bee.